Hi, I'm Padden and I'm part of the Azola group and I'll be one of the student tutors for the Bio-Based Economy module. So why are we so interested in growing Azola? I'm sure you've probably heard some of this before, but it has a massive growth potential up to 35 tons per hectare per year. It's fertilizer independent, which means it will contribute less towards eutrophication of water bodies. It doesn't compete for arable land with food crops, which means there's no competition, which means you can still grow this and insert it into the bio-based economy without reducing the amount of food that's left for other people. And its chemical profile, by which I mean the nutrients in it, as well as some of the fats in it and long chain fatty acids, for instance, makes it interesting as a foodstuff, but also in the production of bioethanol and bio-oil as a substrate. And one thing about azola as well is it can take up pollutants from the environment, which obviously if you're trying to clean water is excellent, but if you're trying to use it as food, it's probably not such a good idea. So some of the problems we have with growing azola are to do with the dissemination storage practices, which have mostly been developed for seed plants historically, not non-seed plants, as non-seed plants have not been farmed historically. So that involves control over sexual reproduction, so we don't know specifically when fertilization occurs, storage potential of reproductive stages, so we don't know the best conditions to store the megasporocarps and microsporocarps in, and how long they will last if we do. There's the reproductive stage maturation process, so if you harvest megasporocarps and microsporocarps early, for instance, will they still mature into and being able to fertilize each other? And then the spalling emergence after fertilization. The other additional problems we have with growing azola are yield stability in the face of abiotic and biotic threats. And this is abiotic meaning more environmental and biotic meaning other life forms. And this is poorly understood in aquatic environments due to most of the focus for the last 10,000 years being on things like corn and wheat, which are land crops, and the low digestibility of the protein-rich biomass by livestock. So it's being shown that azola can only be account for a certain percentage of animals' diets before they start losing weight. So I think in one study, for instance, they showed within sheep could only incorporate 10 to 15% of their diet being azola before they were no longer able, before they started to lose weight. So one way we could get around the current problems we have with growing azola is through the transformation of azola. So transformation of azola would give us a better understanding of the key processes and also the ability to form genetically modified strains with enhanced properties. So what I mean by ability to understand key processes is we currently have an annotated genome of azola and we also have RNA-seq data. So we can say, okay, this strain of azola has less LAR and also produces less polyphenolics, which increased its digestibility. So then at the moment we can look and see, okay, well it expresses more of this gene, for instance, so maybe that's involved in that. So then using genetic transformation we can confirm this by knocking it out and knocking out the gene and seeing what the end product was. And this then also leads on to the ability to form genetically modified strains. So as well as just testing the functions, we could then not only test the functions but knock functions into azola, so that then this sort of next generation genetically modified azola could be the farmed strain. So our current mechanism to transform azola is the agrobacterium mediated transfer of DNA. So initially we were doing this on megasporocarps and microsporocarps whilst they were in the form we call clumps, and this was based on the floral dip protocol. And essentially we were trying to cause the genetic transformation of the Arconogonia cells in the megasporocarp, because these are able to go on to form whole ferns when fertilized by the flagellates of the microsporocarps. And then we modified this according to the Bowertow paper due to they transformed Ceratopteris richardi, which is closer to the fern than Arabidopsis, which is a seed plant. And this used spallings and cut adult materials with digestive enzymes. So they pre-digested it, which formed protoplasts which were more amenable to genetic transformation and also may have undergone dedifferentiation, which then, if you have a dedifferentiated cell that then can go on to form a whole organism, then you can form a whole transformed line, and then we can monitor these transformed lines with GFP signal or with Gus expression. And Gus is essentially just a staining technique that uses a stain that is using an enzyme that generates a stain if the genetic transformation is successful. And we've had some various success with this. So here are some spoolings that we dipped in agrobacterium with the sylvan sucrose. And as you can see on the bottom row, the blue is the Gus staining. So where we see blue, that means we think that tDNA has been taken up by the cells and they have started to express the enzyme which is able to convert the Gus stain. But as you can also get the impression of the amount of spoolings we've had to dip in order to get very, very few transformation events. And we see the same kind of thing here with the adult plant material, various sporadic blue staining. And then the other way that we can look at this is looking for GFP expression, and unlike the Gus staining protocol, this doesn't kill the material we use. So hypothetically, if you observe Gus staining, then you could then have a transformed line. If you observe GFP staining, then you'd have a transformed line. Whilst if you see Gus staining, it means you had a transformed line, but you killed it in order to observe the Gus. But if you can imagine, this is the sort of magnification you've got to look at in order to see GFP, and you've got to look at each plant in at least GFP and RFP to look for background signal in green fluorescence and red fluorescence to make sure that it's not just a background signal. And if you have to do this for 50,000 spoolings and each one takes you 3 seconds, that's going to be a lot of time spent looking at spoolings. So the challenges in transforming the Azolla gametophyte are the low accessibility of stem cells, which lends itself to low transformation rate of the gametophytes. So by low accessibility of stem cells, what I mean is on the left here in the diagram labelled E, you see a megaspore cup with the indusium cap slightly cracked, and the stem cells are inside this, and you can see that in the diagram on the right here with A. So this is a cross section of a megaspore cup, and you can see the developing archegonium, which is what we'd hopefully transform as a site of stem cells. And the reason that we chose this is because we know that the symbiont Nostocozola is passed between the adult plant and the megaspore cup, so we know there is access for bacteria to get in here. But as you can see, it's a relatively difficult to access tissue. The agrobacterium has to move underneath this cap and then get through these layers of tissue. And as you can see from B, there isn't a lot of layers of tissue, so they think maybe in this diagram it's just only one layer of tissue above the archegonium. But still, compared to a callus tissue, for instance, that is relatively inaccessible. So you might think, why don't we just try and increase the accessibility of the archegonium? And so if you see on the right hand picture here, this is the top down image of the megaspore cup, and on the left here, and on the left with the arrow, that's just to give you a frame of reference of how we're, like the angle we're looking at it from. And Yvonne did this, and he chopped off the lid, the indusium cap of a megaspore cup, and these darker ring structures that are slightly greener inside the middle of the megaspore cup, that's oxidative damage, and essentially once you remove the indusium cap, you start to damage the cells inside and they start to die, so we can't just culture them that way. So the next question you say, why don't you just do callus formation? And the thing is that we can't do callus formation in sterile conditions without killing the symbiont, because in order to remove the bacteria or fungus that might grow on it, we'd have to use antibiotics and antifungals, and that might kill our symbiont, which would then not lend itself to the formation of transformed lines, because it would lead to the death of the Azolla plant as well. And so in addition to this, we have little knowledge in how to reprogram somatic cells intergenerative cells without tissue culture. So if we can't form a callus, maybe there's other ways to get somatic cells into generative cells that might be able to form a whole plant without doing this tissue culture. And although there is now some interesting papers about direct dedifferentiation and transformation. And the other problem that we have is the low throughput for observing transformation. So as I mentioned earlier, if you have a gametophyte, a sporling, and you want to measure its expression of GFP, and you have to do around 50,000 in order to, for statistically likely to find one transformant, and each one takes you 2 or 3 seconds, that's a lot of time spent screening sporlings. And then, of course, the other way we have is the Gus staining, and that has the problems we mentioned earlier of just killing your material. So what are the potential solutions for these problems? So alternatively, we could find a highly effective vector or mechanism to transform the azola cells. So that would mean having less throughput. Alternatively, we could identify stem cell markers in somatic plants and use these to reprogram terminally differentiated cells whilst transiently transforming cells. So if we could find a property that made a stem cell-like and put it in a vector and transiently express this in a terminally differentiated cells, maybe we could de-differentiate the cell and thus have a form of generative cells that weren't from the megaspore cup or the microspore cup, and also weren't generated through stem cell culture. And another potential solution would be to increase the throughput for monitoring live transformed sporlings. So if we could get transformation, permanent transformation, then this would be ideal for reducing the amount of time we spent screening. And here's some of the reading that would give you a good background into some of the stuff that I'm trying to do at the moment. Okay, well, thank you for listening to my presentation, and I hope you enjoy the course.